We've started. And there's the beep that tells me we've started. So, where were we? I've said all I'm going to about Trematoda, which is flukes, and Aspidobothrians. We talked a bit last time about the Monogenea. That's all I'm going to say about them. The last group of flatworms we need to cover is the Cestodaria. What, you may ask, is a cestodarian? They're all intestinal parasites of vertebrates. They might go through a life cycle stage that's not in somebody's intestine. Many of them do, in fact. All intestinal parasites. We don't have adult tapeworms in the bloodstream or anything like that, the way we do with certain flukes. The way you tell a cestodarian from a fluke is that the gut is completely lost and nutrition is absorbed entirely through the skin, through that secondary epidermis. Let's see, the basal group of Cestodaria, I actually ought to reorder this slide a little bit, but the very first branch that branches off is called the Gyrocotylidians, and nobody knows jack about them because the only place they live is the intestines of fish in a group called the Chimera, the Chimeras. That's a cartilaginous fish group. Their closest relatives are sharks and rays, but they've been separated from them for 400 million years ago next Thursday. And they have a lot of unique features, and they are almost entirely deep sea fish, meaning that the parasites are not easy to study, and we don't know much about Gyrocotylidians. Amphilydians are a little easier to study, but there's only 8 species of them. And with one exception, they don't parasitize anything that people tend to care about very much. They're parasitic in fish and turtles. So they're also not well known. The biggest group in the Cestodaria by far, the Eucestoda. Remember, EU is Greek for true or good. So the true cestodes are the tapeworms. And the tapeworms you can tell because they've got a body that's segmented. There's a head end called a scolex that's fitted with attachment organs to hang on to the wall of the gut. This could be hooks or suckers or suction cup structures called bothria, or suction cups on stalks called bothridia. We'll see those in a bit. So the head has the attachment and it's divided into are segments. Proglottids are constantly at the rear of the scolex. Old proglottids are constantly so old, mature proglottids constantly pass out the host in the feces. Generally, they're packed full of eggs. So true tapeworms have that body divided into proglottids. Lydians and amphilineid, those other ones don't. No segments there. Real quick, let's look at the basal groups. The outgroup to all of the other Cestodaria, the first group to branch off, is the Gyrocotylidians. They attach to the gut with an oral sucker and a kind of ruffle on the butt called the rosette. They've got ruffly edges as well that probably also increase surface area for attachment. There's no strobila and only one set of gonads, whereas in true tapeworms, you not only have a strobila, a chain of segments, but every segment, every proglottid has gonads in it. Tapeworms have many sets of goads. This is said to be polyzoic, whereas Gyrocotylidians are monozoic. They live in the intestines of chimeras, and there's only one genus with 10 species that's currently accepted as valid. I'd be very surprised if that's all there is. There's probably a whole lot more, But again, this is just not a group that we see very often. I've never seen one. This is just not one that's very widely known. We don't told they actually don't taste very good. We don't commercially fish them for any reason. They tend not to, as far as we know, directly affect humanity very much. We know that the eggs hatch into little spiny larvae called decacanths. Acanthos is Greek for a thorn. Decacanth means 10 spines. And we don't really know what they for the decacanth stage, whether they penetrate into an intermediate host or maybe directly infect a new chimera. So yeah, we don't really understand the life cycle. And here's one of them. The head is at the right in this picture of Gyrocotyle urna. The rosette is at the left, that kind of lumpy, vaguely cauliflower-looking lump over to the left. You can see the ruffles on the sides, although not all gyrocotylids have them. And I came across a paper a while back that actually did try to survey gyrocotylid diversity. So you're looking at a bunch that have the rosette at the lower side of the slide, the bottom of the slide, and the head is facing the top. You can see that many of these are quite roughly, and you can see there's diversity in shape and form. But as far as I know, nobody's really quite, come to terms with this group. I don't think we, I'm not even sure how many species we're looking at here. So that's what they look like. The anatomy we'll go over quickly. There's a sucker at the front, at the top. You can see the rosette at the bottom. The ovaries in the rear to a seminal receptacle where sperm are received. These are hermaphrodites, by the way, but evidently they need to fertilize each other. The eggs are fertilized and then travel down a long folded uterus labeled U. You can see it's got a lot of folds in it, occupying more or less the middle third of the critter, ultimately out the vaginal pore towards the head end. And that's about all we know. There's a chimera, a species of Hariata in the Gulf of Mexico. These are known as chimeras, ghost sharks, or spookfish. And again, they're most closely related to true sharks and rays. They have cartilage skeletons, but they've been separated for 400 million years. There's a number of differences between these and actual sharks. The upper jaw is fused to the brain case, for example. Got funny little pinchers on their heads that the males use to hang on to the females. And they're primarily found in the deep ocean, meaning we don't know an awful lot about most chimeras. There's only a few species that come into the shallows, only a couple that have ever been kept in aquarium. And so we don't know a lot about the parasite's life cycles either.
Dr. Wagner, I'm having a little bit of issues hearing you. You keep breaking up. And I noticed a few other people have said the same thing in the chat.
Okay. All right. I'm not entirely sure what I can do about. I'm not entirely sure what I can do about that. I'll just try to go slowly. We'll get through this. If the lecture is too bad quality to upload, I may have to repeat it on Wednesday.
It's not constantly, it's just off and on.
Okay. All right. I'll go slowly, I'll speak clearly, and if you need me to go back and repeat something, I'll do it. That's all I can think of to do right here.
Okay, I just wanted to let you know.
Okay, thank you. Thank you.
You're welcome.
All right. Okay, moving right along. The outgroup to true tapeworms is also not segmented. They're monozoic, meaning that they're not divided into those proglottids. The body's very flat and kind of leafy. They are parasites in the body cavity of turtles and fish. They've got larvae that live in crustaceans. Only 8 species are known. There is one that parasitizes sturgeon, which are commercially important both for their flesh and for their eggs, known as caviar. The others parasitize things like Australian long-necked turtles, which are elegant and beautiful creatures, but they tend not to get the same degree of attention that other organisms do. And there's a little blood in the next slide. The yellow wormy things crawling around in there are amphilinids, Gigantolina in the body cavity of an Australian long-necked turtle. Those yellow windy things right there, those are the parasites. This is the one from sturgeon. You can see there's a proboscis at one end, but there's no actual gut. Like all cestodarians, they're taking nutrition through the skin, through the tegument. There are yolk glands and testes over to the sides. Most of the winding pinkish stuff in that diagram is female reproductive organs. And that's all I'm going to say about amphilinids. Now we come to the true tapeworms, the Eucestoda, and there's a diagram on the left and a colorized photo on the right of a pretty typical one. Taenia solium is the pork tapeworm. This is the one that you can get from eating undercooked pork. The USDA does try to inspect the meat that's sold in this country, but still, this is why rare pork has never really taken off as a culinary delight, and this is why you're advised to cook your pork pretty thoroughly, because you don't want this growing in your gut. You're looking at the scolex. You can see there's 4 suckers arranged around the scolex, and at the tip of the scolex, there's a little extension called the rostellum, and in Taenia solium, it's armed with a double row of hooks. So you can see the hooks at the very top. Again, because these are cestodarians, food is absorbed straight through the tegument. And different species have different arrangements of attachment organs. So Taenia solium, the pork tapeworm, is at the right. On the left, Dipylidium caninum is the common dog tapeworm. And you can see it's got 4 suckers, and that little bump in the middle of the head is the rostellum, but there's no hooks on this one. So different species, different arrangement. You could get Diphyllobothrium from eating raw fish. And this is yet another species of tapeworm, and this one doesn't have hooks or suckers. What it's got is a pair of kind of leaf-shaped grooves on the scolex that act kind of like elongated suction cups. You can see one clearly at the left. At the left, that's a picture taken with a scanning electron microscope. In the middle, that's a microscope mount that's been stained. On the right, that's an actual specimen taken with a regular camera. And all 3 of them show these suction grooves known as bothria. So Diphyllobothrium means two leaf-like bothria is what that breaks down to. And again, humans sometimes get this from consuming, I think typically raw salmon. This thing was only discovered about 10 years ago. This is a tapeworm in the order Rhinobothridia, which was only named in 2009. And these are parasites of stingrays. And the scolex right here has 4 Bothridia. Bothridia are like Bothria, but they project from the scolex. They're not grooves on the scolex. They stick out on little stalks. So this thing has 4 suction type structures that look like big leaves, but you can see that they're sticking out from the tip of the scolex on little stalks of their own. So those are Bothridia. I just heard something go ding dong, if you'll pardon me. Okay, let's see. Okay, I'm checking the chat. Okay, sorry that I see we're having problems with me breaking up. If it gets really bad, let me know. I'm just trying to go slowly and be as clear as I can. And if I have to repeat any of this stuff, I can. Let me know if it gets impossible to deal with. Okay, right. I see you. You're here. Okie doke. Let's resume. Some tapeworms have added accessory attachment structures as well. This one's from Skates and Rays that has 4 retractable barbed tentacles connected to its Bothridia. So this one looks really unpleasant to have up your gut. Fortunately, I'm not a stingray. And there was a recent attempt to document tapeworm diversity more fully, looking not only at the species that are of commercial, medical, or veterinary importance, but look at a wider range of hosts than ever before. They counted almost 5,000 species, including 215 that were new, that nobody had seen before. Up at the top, that's Yorkshire Izzardi, which has Bothridia with hooks sticking out of them. And then on the right, Litobothrium nicolai just looks like something I'd hang on my Christmas tree. You can see it's got some Bothria. Those are those leafy things at the widest part. But yeah, the whole thing just looks really strange. There's a lot of tapeworm diversity out there, and a lot of it has not been studied well. Anyway, that's a complete tapeworm in genus Echinococcus. This is a pretty common parasite of carnivores. Dogs and cats could get this. Humans will sometimes accidentally ingest the eggs. And as we'll see, either today or Wednesday, eating Echinococcus eggs can damage humans pretty badly. Although we don't get infected by the adults. We can get infected by a type of larvae that I'll get to later. I think I was reading about this, that one way that humans get infected is if you have infected carnivores like dogs or foxes defecating in your garden. Eggs can get washed out of the feces onto your produce. So you do want to wash everything before you eat it, of course. But anyway, Echinococcus at the right, you can see the scolex. You can see the suckers. Those 2 circles that look like eyes are in fact suckers. And the strobila here only consist of a few proglottids. I count 4. At the far left, that one is the most mature, and that is about ready to break off, and the proglottid and the eggs in it will pass out of the gut and the feces. So the rightmost one, or the leftmost one, is just about ready to go. Then you have two, and then I see the fourth is only beginning to be defined at the back end of the scolex. So new ones appear at the end of the scolex, and as they mature, they get pushed down the chain, and at the end of the chain, the oldest ones, once they're mature, break off. So this thing is constantly budding off new proglottids and losing the old ones. And this Echinococcus has four. This Taenia saginata, that's the beef tapeworm. It's not a it's not it's called a beef tapeworm, not because it lives in cows, but because you can get it from eating larvae in beef. This one consists of thousands of proglottids. Now you can see why these are called tapeworms, because it, well, duh, looks like a tape. Okay, and yeah, by the way, the scolex is up at the top of the picture, and you can't even see any detail on it. Little look at the anatomy. The tegument follows your basic neodermate pattern. Below the muscle layer, you have the cytons, the cell bodies. Those are down at the lowest part of this diagram, the cytons, or as they're called, pericarya. They send up these branches, these processes that pass through the muscle layers, in between the muscle cells. And I should mention that the muscles on some of these are fairly well developed. Tapeworms can be surprisingly active. And those processes go up through the muscle layer and then fuse and join to form that tegument, that syncytial layer. Unique to the true tapeworms is the fact that on the outer side of that syncytium, you have a whole bunch of microvilli. Remember that a microvillus is a little finger-like, more or less permanent projection from a cell. And you can see a whole bunch, looks like the teeth of a comb at the very top of this diagram. They're called microtriches. If you just have one, it's called a microthrix. So all of those comb teeth at the very top are microtriches. These increase surface area. These enable tapeworms to feed more efficiently because, remember, all of their food is coming through that tegument. They also probably increase adhesion. They enable the worm to adhere better to the gut wall. It's not that easy making your living inside a gut. It's kind of like living inside a tunnel, right? If you're living inside a tunnel, the oncoming traffic tends to push you in one direction, and the same is true for gut parasites. They've got a tendency to be pushed out by the natural peristalsis of the gut and the passage of feces. So all those hooks and things certainly have a reason, and the microvilli probably help the tapeworm to adhere just as well. Live tapeworms, if you look carefully, have an almost velvety appearance to them, and it's because of all of the microtriches. Microtriches help anchor the worm. They increase the surface area, and depending on the species, they can take different shapes. This particular one has two types, very slender, hair-like ones. Each hair-like one is called a philothrix, and these flat, spiny, scale-like ones are called spinothrix, which sound like the heroes of an Asterix comic book. Asterix and Obelix's best friends are Philothrix and Spinithrix. Not everybody's going to get that reference. But yeah, the shape is important for classification. There's other types than this. I'm not going to say much more about it. But if you study tapeworms, this is one of the things that you'll end up encountering. Okay. Like most parasites, tapeworms have their reproductive capacities cranked up to 11. This is a mature proglottid of Taenia solium, the pork tapeworm. The male reproductive organs are in blue, of course, and the female organs are in pink. And you can see that the ducts of both lead to a little chamber called the genital atrium, which opens to the outside world through a genital pore. Tapeworms can self-fertilize if necessary. Many of them can, but they'll pair up as well. If you have two of them in the gut, they'll crawl together and put their genital pores together, and each one will fertilize the eggs of the other to trade. Typically, male organs develop first, and they'll store sperm, and then when the female organs develop, they'll either release the sperm or they'll retain them and fertilize their own eggs. That's probably to prevent self-fertilization, although, as I've said, these can self-fertilize if they need to. So the male organs typically consist of many testes. Each one of those blue dots is a testis. Each testis has a vessel connected to it called a vas efferens. And the vasa efferentia, all of those vases efferenses, join together to form a common tube called a vas deferens. That's that thicker kind of winding tube that leads right to the genital atrium over on the far right. So there we've got the testes, or as I like to tell my students in this class, I am the tester, and that makes you, well, never mind. Female organs typically consist of an ovary, and in this genus, it's branched. So if you look at the pink stuff, there's that round pink thing just a bit up from the center. The branching things to left and right of that are ovaries. The eggs pass through, actually I take that back, the ovaries up at the top, I misspoke. The eggs, no, I take it back. The ovaries are at the left and the right. They pass through the oviduct into that central structure called Melis's gland. They're covered by yolk cells produced in the vitellaria, which is up to the top, and they're stored in that sac hanging down. That's the uterus. So there we go. Eggs are to the left and the right. The yolk glands are up at the top. Melis's gland in the center is where the yolk is added and the shell gets added, and then the eggs are stored in the uterus, which is that kind of club-shaped pink thing hanging down. As I've said, many tapeworms are hermaphrodites and can self-fertilize, although they often prefer to outcross. There's at least one, actually there's several, that have separate sexes, and this is one of them. This one's a parasite of shorebirds, seagulls and things like that. Usually this one has separate sexes. Usually when you find these, there'll be two in a bird, one male and one female. This probably means that sex determination is influenced by the environment, that possibly the first worm to colonize becomes a female, and then the second one that comes in is stimulated to develop into a male. So sex determination may depend on environmental triggers released by maybe the first worm to get in there. I'm not quite sure how that works out in practice. It would be an interesting question to look into. But certainly most tapeworms are hermaphrodites. And as the uterus fills up with eggs, everything else degenerates. And that uterus, which was a simple sac, can take on rather complicated branching shapes. And this is important for identifying species. If you're doing a fecal slide, you'll often find proglottids in the feces, and you may need to identify what species you've got by looking at things like uterus shape. So at the top, that's the uterus of Taenius solium, the pork tapeworm. Below, that's the uterus of Taenia saginata, the beef tapeworm. And gravid proglottids are the ones that constantly break off. In some species, they may break down in the host gut and the eggs are released and the eggs travel out separately. Or the proglottids, or even whole chains of proglottids, may pass out of the host body whole and sometimes still moving. Years ago, we briefly adopted a puppy. He did not really work out. We sent him to a farm. Actually, we really did send him to a farm. People always look really uncomfortable when I tell him that, but my wife actually knew somebody who really did live out in the country and have a farm. So hopefully that dog is very happy now. When we got him, he had tapeworms. We had to take him to the vet. And I had the unique pleasure of cleaning up after this puppy and having to clean up fresh deposits on the floor that still had little wriggling things in them. Those were chains of proglottids of the tapeworm, whatever it was that this dog happened to have. So entire chains of proglottids can break off, pass out, and may still be alive and kicking or alive and wriggling outside of the dog's body. Ew. Other species, however, you never see that because the proglottids break down and the eggs, which are microscopic, are released to travel through the body independently. The embryos develop within a thin membrane called the embryophore. What you're looking at here, the actual embryo is that central part, and they're out of focus, but if you look closely, you can see a couple of the 6 little spines that the embryo will use. Tapeworm embryos like this are called hexacanth larvae because they'll have 6 thorns or 6 spines to help tear their way out. So that central circle is the actual embryo. It's surrounded by a thin membrane called an embryophore. Outside of that, you have the inner envelope and the outer envelope. And surrounding the whole thing is the eggshell-like coat. So the coat is that darker brown layer that you see. The inner and the outer envelope, it's kind of hard to tell the distinction. Well, I guess I can see it. The outer envelope is a little bit darker. The inner envelope is actually derived from cells from the embryo itself. The outer envelope is derived from a cell in the parent called a vitelline cell. And you'll see eggs like this in a forthcoming lab. You'll see this very species, in fact. In the time we've got left, let's look at the life cycle, and we may not finish this. The life cycle can be very variable, but as we said, the adults live in the gut. Eggs and or proglottids pass out of the definitive host in the feces. In a few species, the eggs hatch into ciliated larvae called chorocydia, which are closely analogous to the miracidia larvae that we saw in the flukes. and those may get swallowed or may penetrate an intermediate host. More commonly, the eggs get swallowed by an intermediate host, and the eggs never hatch into a swimming larva. They hatch into a larval type called an oncosphere. Onco means a hook. Sphere means a sphere. They're basically sphere-shaped larvae that have little hooks that they can use to tear their way through the body. They look a little bit like Jigglypuff crossed with Freddy Krueger, right? Little sphere-shaped thing with little microscopic, wicked little hooks clawing their way through the body. Yeah, Jigglypuff meets Freddy Krueger is about the best way I can describe it. The oncosphere larvae will travel through the body of the intermediate host and form a stage called a metacestode. And there's actually many types of metacestode that's out there. When the definitive host eats the intermediate host, the metacestodes transform into adults, and round goes the great circle of life. Brace yourself for the next slide. That's a drawing from Japan from about the year 1850, showing a man who's been eating raw salmon. You can tell because a sizable chain of proglottids has developed in his gut of a species called Diphyllobothrium nihonkaiense. This is one of the species where the proglottids can pass out still in considerably long chains, as you see them being extracted from the man's gut. What the woman is doing, sitting and pointing, I'm not entirely sure. She's either directing operations or just laughing. I should mention, by the way, you can get Diphyllobothrium from eating raw salmon. This tends to be a problem in Japan where the sushi is extremely fresh. In the United States, the official rule is that any fish that's going to be used for sushi is supposed to be flash frozen. Usually they do this right on the boat. The boat will contain a freezer with plenty of liquid nitrogen, and they'll instantly freeze the fish. And if you flash freeze, it's not supposed to have too much of an impact on taste, although really it probably does a little bit, but most people aren't connoisseurs of this sort of thing. And flash freezing for 72 hours should kill the infective stage. But if you eat really raw fish, my wife's father was a commercial fisherman, and he used to get on the boat with his soy sauce and wasabi right there. And he got sushi that was, still flopping on the deck. He really liked it. And I never heard if he had any problems with parasites, but something like that could conceivably have put him at risk. The U.S. fish supply is mostly pretty safe, unless you find a restaurant that's maybe right on the coast and has access to very fresh seafood that they don't freeze. So this is not too likely to happen to you unless you indulge in very fresh, delicious sushi. Okay, sorry about that. So the proglottids or the eggs pass out. The eggs sometimes hatch into ciliated larvae, but usually they hatch into oncosphere larvae, which are also called hexacanth larvae. And this is one. You can see individual cells in the hexacanth. Up at the top, there are a couple of cells that secrete enzymes and form a penetration gland. That secretes enzymes that digest the host tissue. And between the enzymes from the penetration gland and those 6 hooks, see what I mean about how this thing looks like Jigglypuff Freddy Krueger? Those hooks and the enzymes enable the larva to tear its way out of the intermediate host gut and enter the body cavity or find its way to a major organ. As I've said, there's a few that will hatch into a choroncidium that's eaten by an intermediate host. On the left, you've got a Coracidium, but the hexacanth is beginning to emerge right where you see that arrow.
I'll send you a text with apartment information.
Okay, I love you, sweetie.
Oh, I do. He's asleep.
Oh,
He has a test when he wakes up and he needs to shower.
okay, got it.
He shouldn't do anything else until he gets the exam.
I'm on it, but he can sleep as long as needed.
Oh, he seems exhausted.
Okay. Okay, right. Evidently, my kid has a test. Okay. The Coracidium gets eaten by an intermediate host, and then the hexacanth emerges and shucks that ciliated epidermis and leaves it behind. You can see two stages in the process here. In species with a Coracidium, the hexacanth develops into one of these metacestodes called a procercoid. This is a copepod, a little swimming aquatic crustacean with a procercoid developing inside it. And then when the intermediate host gets eaten by a the procercoid develops into a plerocercoid. And this is how you get tapeworms coming out of your butt. This is the life cycle of Diphyllobothrium. Eggs pass in the feces, the Coracidia hatch, and they're eaten by crustaceans. Inside a crustacean, the Coracidium becomes a procercoid. When the crustaceans are eaten by freshwater fish, the procercoid is released from the crustacean and develops into a plerocercoid. The plerocercoid then might pass into a predator fish. The plerocercoid might get passed up the food chain until a human finally eats raw or undercooked fish that's infected. And if a human eats the plerocercoids, they develop into adults in the intestine and start releasing eggs. And it's the circle of life. We're going a little long, but one last slide. In species that don't have an aquatic stage like that, the hexacanth, that Jigglypuff Freddy Krueger larva, will dig its way through the gut and may end up in an organ or may end up, in this case, in the muscle. And in the case of pork tapeworm, the larvae develop into an infective stage called a cysticercus. Down below, that's a whole cysticercus, and you can see that strongly staining spot. That's a new scolex. And that scolex is tucked inside the cysticercus. And when the cysticercus is swallowed, the scolex will just pop right out, latch on to the definitive host intestinal wall, and grow into a brand spanking new tapeworm. Up at the top, you've got meat that's infected. All of those little white round things are cysticerci. What happened was that this is a parasite of humans. Eggs passed out in human feces. Pigs ate the eggs. This tends to be commonest in places where human waste is used as fertilizer, where pigs have an opportunity to root through, human feces or, feces contaminated soil or something like that. Ever heard the expression happy as a pig in shit? Well, yeah, they're not really happy, because if they root around in it, they can swallow eggs. The eggs hatch in the gut. The hexacanth larvae tear their way through the pig's gut and form cysticerci in the muscle. This is referred to as measles. Meat inspectors will look for measled pork, pork with these little white spots in it. And yeah, those are cysticerci, and that meat is not safe to consume. Although, be it said, cooking your pork thoroughly will kill off any stray cysticerci that are present. You wouldn't actually get this unless you happen to be eating raw pork or possibly some other food that might have been contaminated by pork. I remember reading an account. There's a book called New Guinea Tapeworms and Jewish Grandmothers, written by a parasitologist, a medical parasitologist who met a very sweet Jewish lady who kept very strict kosher, but she had pork tapeworms. And the reason was the butcher shop where she was buying her fish was also butchering pork and not keeping their utensils clean between times. So the meat and the fish that she was buying was getting contaminated with pork, and she actually got Cestodaria in her food, even though she wasn't eating pork. She would have been horrified too. All right, that seems to be what I got time for. So I'll go ahead and break it off there, although there's more to cover. I'm not quite done. The slide handout is up if you're a undergrad. It's up on Blackboard, and it will be up for the grad students shortly, as soon as I can get it uploaded. And you'll get a little sneak preview of some of what we will cover tomorrow, but we'll pick that up tomorrow, and I'm going to stop recording.